Hi, I'm Patrick Frantom. I'm a professor in the Department of Chemistry and Biochemistry here at the University of Alabama, and I'm excited to have the opportunity to tell you a little bit about the work that we do in my lab. My lab is in the Biochemistry division of the department, and as such, we're mainly interested in structure-function relationships in biomolecules, and most specifically proteins. So my lab has continued, kind of my training is in protein chemistry and mechanistic enzymology, and we've really continued those kinds of investigations in my independent career here at Alabama. That the kind of the big question that really drives our core research is how do proteins do all of the wonderful and amazing things that they do? And so you can think about enzymes as these amazing catalysts. You can think about regulatory strategies that proteins use to turn on or turn off their activity. And so we're really interested in understanding how this, polymer of 20 amino acids is able to do these really interesting functions. We come at this through a number of different ways. We use recombinant expression and purification in order to basically isolate the protein of interest. And so we really study that protein as a molecule into itself and characterize the function of that molecule. We use a lot of enzyme kinetics when we look at how well an enzyme does the reaction that it is supposed to do. We also look at things from a structural perspective, and one of the techniques that we like to do for this is a technique called hydrogen deuterium exchange. This allows us to probe for subtle conformational changes in the protein structure under solution chemistry rather than in a crystal structure. And I'll tell you a little bit more about that later, but we use that as a technique that really complements nicely X-ray crystallography, where we tend to do most of our work with Dr. Dunkel, who's also in the department. Our two current projects that we're working on in the lab, one of them is focused on proteins that are involved in the formation of iron-sulfur clusters, and I'll show you more about those in a minute. The other protein is looking at sugar chemistry and enzymes that catalyze the formation of glycosidic bonds called glycosyl transferases. So let's take a look at some more details about these proteins that are involved in iron-sulfur cluster formation. So this is a slide that shows the family of proteins that we work with which are involved in iron-sulfur cluster biosynthesis. You can see three examples of iron-sulfur clusters in the upper left-hand part of this slide. Iron-sulfur clusters are essential cofactors that are used for redox chemistry in all living organisms. You're probably most familiar with iron-sulfur clusters either from studying aconitase in the citric acid cycle or from proteins that are involved in the electron transport chain in mitochondria. All of these proteins require iron-sulfur clusters to pass along electrons and do redox chemistry and keep cells functioning properly. One of the challenges of building these iron-sulfur clusters, too much sulfur and too much free iron is actually toxic for those systems. So to build these clusters, their assembly actually has to be very tightly regulated so that these toxic building blocks don't get out and cause other damage. So for this SUF pathway, which is a pathway which is found in bacteria, it's actually a combination of numerous proteins that play roles, and each step in this assembly has to be choreographed. The first step in this reaction is catalyzed by an enzyme called SUF-S. You can see that is shown in the blue ribbon structure. SUF-S is responsible for mobilizing the sulfur that's going to be used to build the iron-sulfur clusters. It pulls the sulfur off of cysteine and transfers that sulfur to an accessory protein called SUF-E. And you can see that in the green ribbon diagram. SUF-E then passes the sulfur onto a larger protein complex called the SUF-BCD complex. And you can see that in the cyan, pink, and yellow ribbon structure on the right-hand side of this figure. This is the actual scaffold protein that's going to build the iron-sulfur cluster. Iron is mobilized to this enzyme. We don't know where the iron actually comes from in the cells. That's still a little bit of a mystery. But ultimately, an iron-sulfur cluster is made on the SUF-BCD scaffold. And then once the mature cluster is made, it gets transferred to a SUF-A protein that then goes and targets the iron-sulfur cluster to its downstream target proteins like aconitase. So we've got this nice schematic here that shows kind of the basic role for each protein, what its job is. What we really don't understand at this point are molecular mechanisms of how each protein in this pathway is actually doing the job it's supposed to be doing. And so we're looking at trying to isolate these proteins and characterize their function and try and identify structure-function relationships in each of these proteins in this pathway. Now, why do we want to do this? Why are we interested in this? And why is the NIH funding us to do this? Well, two reasons. Again, this is an essential pathway, and there are some parallels between this bacterial pathway that we study and the human pathway. Problems in the human pathway lead to several human diseases, so we're interested in understanding overall how these different pathways function. But more importantly, in the bacterial pathway, there are a number of bacteria that this is their only mechanism for making iron-sulfur clusters. And so if we can understand better the function of each of these proteins and how they're working at the molecular level, then we're in a much better position to design small molecules that could disrupt their function and be potential antibiotics that would be targeting a brand new system. Now, again, one of the ways we look at these proteins are that we characterize these proteins or to recombinantly express them in E. coli and purify them, and then look at their function. And we do this through a wide range of kinetic assays. The other really interesting way we look at these proteins is to try and understand something about their conformational dynamics. What are they doing in solution? Right? And what you can see, the pictures you see on this picture are static crystal structures. So in general, you can think of these as snapshots, but we know proteins in solution are actually very dynamic. They're going to be sampling lots of different states. And in this particular family of proteins, we think that coordination of these different conformations is actually what helps to choreograph the pathway. For example, if you look at the first step where the sulfur is transferred to SUF-BCD, you see that SUF-BCD has to both interact with SUF-E, the green protein, and when it has an iron-sulfur cluster built, it has to interact with SUF-A, the red protein. We don't understand at all how SUF-BCD knows when it's supposed to interact with SUF-E and when it's supposed to interact with SUF-A. Our hypothesis at the moment is that perhaps there's a subtle change in conformation between the SUF-BCD protein without the iron-sulfur cluster and the SUF-BCD protein with the iron-sulfur cluster. In order to try and understand what those changes in conformation might be like, we're exploring crystallographic experiments to try and solve crystal structures. But as a complement to that, again to these static structures, some of these changes are probably changes in the protein motion. And so we need a different experimental technique to look at that. And the experimental tech we use is backbone amide hydrogen deuterium exchange followed by mass spectrometry. And that's a really big name, so we abbreviate it as HDX-MS, but the fundamentals of this technique are actually pretty straightforward. The way that this technique works is we take a protein and we incubate it in D2O for various amounts of time. Now in the figure here you can see that this protein, what we've labeled are the amide backbone nitrogens of each of the peptide bonds in this protein. And each of those amides has a proton, and that proton will exchange with D2O for a deuterium. And you can see that on the right-hand side by the red dots that have exchanged with blue dots. Now the rate of exchange is dependent upon the hydrogen bonding of those amides and the solvent accessibility of those amides. So if you were looking for a technique to measure conformational changes, you would want something that's going to be sensitive to solvent accessibility and changes in hydrogen bonding. Since a deuterium is one mass unit heavier than hydrogen, we actually can measure the change in mass of the protein as it collects more deuteriums. To show you the experiment in a little more detail, we put the protein again into D2O. We can see it exchange over certain amounts of time, and we vary the amount of time the protein is in the D2O. At various time points, we quench the reaction by going to low temperature and low pH, and that stops the exchange between the protons and the deuterium. We then digest the protein into various peptides that we separate using HPLC. As those peptides are separated, we flow them into the mass spectrometer, and we can get a mass for each of those peptides. Now, if we follow each individual peptide at various time points, what you see is a figure that the figure on the right-hand side of this graphic, in that the mass envelope of this specific peptide, going from water all the way up to 3 hours of incubation in D2O, the mass increases of that peptide. And so what we can do is measure the increase in mass of a peptide under different conditions. And for example, for SUF-BCD, we can measure the changes in mass in the presence of the iron-sulfur cluster or in the absence of the iron-sulfur cluster. And that can start to give us an idea of what parts of the protein might be changing conformation. Once we've identified those parts, we can then go back and use our mechanistic enzymology approach to understand what the function of those parts are. So we really use this structural technique of HDX-MS in order to give us hypotheses that we can then test using mechanistic enzymology approaches. So that's a really brief introduction of the type of research we do. I didn't want to go into too much data and things like that, because I think that tends to kind of bog down in the initial introduction to a lab. But I do think one of the important things to do, especially since this is going to be a virtual visitation weekend, is let's actually go see the lab. So let's go down the hall and check out the lab. All right, welcome to the lab. We're still under pretty strict COVID restrictions in terms of how we can work in the lab. So I've got my mask in place here. And I want to show you a few of our favorite toys. So one of them is our HPLC unit. So we use this unit for looking at quantitating enzyme products that are made, especially products where we can't necessarily get a UV vis signal to use them on our regular spectrophotometers. This one really helps us out in that case. This is really nice. We've got an auto sampler, so we can set up to 96 reactions at once and let it push go and forget it, which is really nice. And then we've got a number of different detectors as well. So we have a fluorescence detector as well as a photodiode array, which is able to look at the full wavelength spectrum of whatever is coming off of the HPLC. Let me move you over here to our next one. Let's see how this is pretty good. Give you the rolling tour. So another favorite toy, and you'll hear the buzz of the cold box here, is our new automated FPLC system. So this is the system that we use in order to purify our protein. We've got a number of different chromatography columns that we can use to separate proteins based on different physical or chemical properties. Again, the nice thing about this is that once you've grown your E. coli and lysed the cells, all of this is automated. So we put in our sample, hit go, and we're able to collect our fraction collector over here and get our purified protein off of the different columns that we use. So a lot of experience, our students get a lot of experience using the FPLC and learning how to purify recombinant proteins. Then the last two toys that I'll show you are our spectrophotometers. It's hard being the cameraman and the presenter. So we have two Carey spectrophotometers, and this is what we use to do our enzyme assays. It's important to have two because when you have a lot of people in the lab, sometimes those can become the rate limiting step. And so those are our main workhorses that we use to look at enzyme kinetics and how either mutations to enzymes or different assay conditions can affect the functional properties of different enzymes. Now, the last thing I want to do is take you downstairs for a minute and show you the mass spectrometer setup that we have for doing that special HDX-MS technique. All right, this is our
fairly new Zevo Waters mass spec time of flight mass spectrometer. So we have this spectrometer interfaced with a very specific LC system, which is specifically designed for doing hydrogen deuterium exchange mass spectrometry. Basically, everything that I showed you in the little schematic about how you do HDX, really after you quench the reaction, all occurs in this LC system. So it has a protease column inside that actually will digest the protein into peptides. It has the C18 column inside that separates those peptides and then sends them directly into the mass spec for analysis. Now, for each of these proteins that we look at, we're usually looking at somewhere in the neighborhood of 100 to 200 peptides that we're analyzing to follow the amount of deuterium put into those peptides over time. Importantly for that, we've got some really nice software upstairs that automates that process and works really well with the mass spec, or the data that's collected on the mass spec, in order to streamline that process. Okay? Well, let's head back upstairs.
Okay, well that just about does it for our whirlwind virtual tour of the Frantom Lab. I'd be happy to talk with anyone in more detail about the research we do. Like I said, I kept it pretty brief in this. Hope that I'll get to meet and speak with some of you at our virtual recruiting weekend coming up. Thanks for watching.